Targeted Therapeutics, Oligonucleotide Conjugates for Transfection. Narrated by Luke McLaughlin, biotech digital marketer, business developer, and life science content creator. The conjugation of oligonucleotides with various ligands to enhance their delivery, stability, and specificity for targeted therapeutics has become a cornerstone in the development of next-generation molecular therapies. These ligands can facilitate targeted delivery to specific tissues, cells, or intracellular compartments, thereby improving the therapeutic index of oligonucleotides. Beyond cholesterol, folic acid, and spermine, a wide range of ligands have been explored for these purposes. Here's an overview of the classes and examples of ligands used in oligonucleotide conjugation: peptides and proteins, cell-penetrating peptides, CPPs, short peptides that can cross cell membranes. Examples include TAT from HIV TAT protein, penetratin from Antennapedia homeodomain, and R9, a nona-arginine. Cell-penetrating peptides, CPPs, also known as protein transduction domains, PTDs, are short peptides that facilitate the delivery of various molecular cargoes across cell membranes. These cargoes can range from small molecules and peptides to larger molecules like proteins, nucleic acids, ttDNA, RNA, and even nanoparticles. CPPs have garnered significant interest in the field of drug delivery and molecular medicine due to their potential to overcome one of the major challenges in therapeutic development: efficient cellular uptake and bioavailability of biologically active compounds. Characteristics of CPPs. Size and composition. Typically 5 to 30 amino acids in length, CPPs can be cationic, positively charged, amphipathic, containing both hydrophobic and hydrophilic regions, or hydrophobic. Mechanism of entry. CPPs can enter cells via endocytosis or by direct translocation across the plasma membrane, depending on the peptide sequence, cargo, and experimental conditions. Cargo attachment. Cargoes can be covalently attached to CPPs or form non-covalent complexes for delivery into cells. Mechanisms of cellular uptake. Direct penetration. Some CPPs can directly translocate across the cell membrane through a mechanism that is still not fully understood, but may involve transient formation of inverted micelles or other disruptions of the lipid bilayer. Endocytosis. This is a more commonly accepted pathway where CPP cargo complexes are internalized via various endocytic pathways, including clathrin-dependent, caveoli-dependent endocytosis, and macropinocytosis. Types of CPPs. Primary cationic CPPs, rich in basic amino acids like arginine and lysine. For example, TAT peptide from HIV-1 TAT protein, arginine-rich peptides. Secondary amphipathic CPPs. These peptides form amphipathic structures either in solution or upon interaction with membrane lipids, for example, transportin. Tertiary hydrophobic CPPs, primarily composed of hydrophobic amino acids. Applications: drug delivery. CPPs can transport therapeutic agents into cells, including chemotherapeutic drugs, proteins, and gene editing tools, like CRISPR-Cas9 components. Molecular imaging. CPPs tagged with fluorescent dyes or radiolabels can be used for imaging and diagnostic purposes. Vaccine development. Enhancing the delivery of antigens or adjuvants into immune cells to improve vaccine efficacy. Advantages. Versatility. CPPs can deliver a wide range of cargoes into different cell types, including hard-to-transfect cells. Low toxicity. Many CPPs exhibit low cytotoxicity, making them suitable for therapeutic applications. Ease of synthesis. CPPs can be synthesized relatively easily and can be modified to improve their properties or functionalized for specific cargoes. Challenges and considerations. Endosomal escape. Ensuring that the CPP cargo complex escapes the endosome to reach the cytoplasm or nucleus remains a significant challenge. Specificity. Many CPPs lack cell-type specificity, leading to non-selective uptake by cells and potential off-target effects. Stability and immunogenicity. Stability in biological fluids and potential immunogenicity of CPP cargo complexes need careful consideration. Future directions. The field of CPP research is evolving, with ongoing efforts to improve the specificity, efficiency, and safety of CPP-mediated delivery systems. Advanced strategies include the development of stimuli-responsive CPPs that activate under specific conditions, for example, pH-sensitive, light-activated CPPs, and targeting motifs to direct CPPs to specific cell types or tissues. As our understanding of CPP mechanisms and cell biology deepens, CPPs hold promise for revolutionizing the delivery of therapeutics and diagnostics, potentially overcoming some of the most significant barriers in modern medicine. Antibody fragments, such as single-chain variable fragments, scFv, targeting specific antigens on cancer cells. Antibody fragments include various forms of antibodies that retain the antigen binding domain, but lack the Fc region of the full antibody molecule. Common types include Fab, fragment antigen binding, a monovalent fragment consisting of one of the arms of the antibody, including the light chain and part of the heavy chain. scFv, single-chain variable fragment, comprises only the variable regions of the heavy VH and light VL chains, linked together by a short peptide linker. Fab-sent, 2, similar to Fab but dimeric, retaining both antigen-binding arms. Targeted delivery. The primary role of antibody fragments in transfection is to achieve targeted delivery of nucleic acids. Specificity. Antibody fragments are engineered to recognize and bind to specific cell surface markers. This specificity allows for the targeted delivery of nucleic acids to desired cell types, reducing off-target effects and enhancing the efficiency of gene expression or silencing in the target cells. Conjugation with carriers. Antibody fragments can be conjugated to vectors or carriers, like liposomes, nanoparticles, or viral vectors, that contain the nucleic acids. This conjugation can direct the cargo to specific cells through the antigen-antibody interaction. Advantages: increased efficacy. By enhancing the uptake of genetic material by target cells, antibody fragments can increase the efficacy of transfection. Reduced toxicity. Targeted delivery minimizes interactions with non-target cells, potentially reducing cytotoxicity and other adverse effects. Versatility. The approach can be adapted to different cell types and conditions by selecting appropriate antibody fragments. Challenges. Complexity. Designing and producing targeted delivery systems with antibody fragments is complex, requiring sophisticated biotechnological methods. Stability and delivery efficiency. Ensuring the stability of the conjugates and efficient delivery of the nucleic acid cargo into the target cell's cytoplasm or nucleus remains challenging. Cost. The production of specific antibody fragments and their conjugation to delivery systems can be costly. Small molecules. GalNAc, N-acetylgalactosamine, targets the asialoglycoprotein receptor, ASGPR, on hepatocytes, making it especially useful for liver-targeted therapies. N-acetylgalactosamine, GalNAc, is a targeted delivery system specifically designed to enhance the cellular uptake and therapeutic efficacy of oligonucleotides. This system exploits the natural liver-specific uptake mechanism through the asialoglycoprotein receptor, ASGPR, a receptor abundantly expressed on the surface of hepatocytes. The use of GalNAc for transfection is a testament to the ongoing efforts in developing more efficient and targeted delivery methods for therapeutic molecules, particularly for the treatment of liver diseases and disorders. Mechanism of GalNAc-mediated delivery. ASGPR targeting. ASGPR is a hepatocyte-specific receptor that mediates the clearance of glycoproteins from the blood. GalNAc moieties can specifically bind to ASGPR, making them ideal ligands for liver-targeted delivery. Receptor-mediated endocytosis. Once the GalNAc conjugated oligonucleotide binds to ASGPR, it is internalized into the hepatocytes through receptor-mediated endocytosis. Endosomal escape and release. After internalization, the therapeutic oligonucleotide needs to escape from endosomes to reach its target within the cell, such as the cytoplasm or nucleus. Advantages. High liver specificity. GalNAc conjugation significantly enhances the uptake of therapeutic oligonucleotides by hepatocytes, reducing off-target effects and increasing therapeutic efficacy. Reduced dose requirements. Enhanced delivery efficiency allows for lower doses of therapeutic oligonucleotides, potentially reducing the risk of side effects. Broad application. The GalNAc delivery system can be used with various oligonucleotide therapies, including siRNA, mRNA, ASOs, and potentially CRISPR-Cas9, offering a versatile platform for liver-targeted therapies. Challenges. Limited to liver targeting. The specificity of GalNAc for ASGPR means it is primarily suitable for liver-targeted therapies and may not be applicable for diseases affecting other organs. Endosomal escape. Efficient endosomal escape remains a challenge to ensure the release of the therapeutic oligonucleotide into the cytoplasm. Synthesis and conjugation. The synthesis and conjugation of GalNAc to oligonucleotides require sophisticated chemical processes, which can affect the scalability and cost of the therapy. Vitamins, such as biotin, vitamin B7, or vitamin E, which can be recognized by specific receptors on cell surfaces. The use of vitamins in transfection leverages their natural cellular uptake mechanisms to enhance the delivery of genetic material into cells. Vitamins, essential nutrients required for various physiological functions, can also act as ligands that target specific receptors on cell surfaces. By conjugating vitamins to nucleic acids or to vectors carrying nucleic acids, scientists can improve the specificity, efficiency, and safety of gene delivery. This approach has been particularly explored with vitamins such as vitamin B12, folic acid, vitamin B9, and vitamin E, among others, due to their roles in cellular metabolism and their natural receptor-mediated uptake pathways. Vitamin B12, cobalamin. Mechanism. Vitamin B12 is essential for nervous system function and blood formation. It utilizes intrinsic factor-mediated endocytosis for cellular uptake, making it a potential target for delivering nucleic acids to cells. Application. While its use in transfection is less common compared to other vitamins, vitamin B12 conjugation has been explored for targeting specific cell types that express high levels of B12 uptake mechanisms, potentially including cancer cells. Folic acid, vitamin B9. Mechanism. Folic acid targets the folate receptor, FR, which is overexpressed in several types of cancer cells. The high affinity of folic acid for its receptor allows for receptor-mediated endocytosis, facilitating the targeted delivery of the conjugated molecules into cells. Application. Folic acid is widely used for targeted drug delivery and transfection in cancer research and therapy, aiming to increase the uptake of therapeutic nucleic acids by cancer cells while minimizing impact on normal cells. Vitamin E. Tocopherol. Mechanism. Vitamin E is a lipid-soluble antioxidant that is incorporated into cell membranes. Its conjugation to nucleic acids or vectors can enhance membrane fusion or endosomal escape, improving the delivery efficiency of genetic material. Application. Although less directly involved in receptor-mediated endocytosis, vitamin E conjugation has been used to facilitate the transfection of nucleic acids by enhancing lipid-based delivery systems. Advantages. Targeted delivery. By exploiting natural cellular uptake pathways, vitamins can provide a means of targeting specific cell types or tissues, thereby increasing the specificity and efficiency of transfection. Reduce toxicity. The use of vitamins as part of a delivery system can potentially reduce the toxicity and immunogenicity associated with other transfection agents, leading to safer gene delivery methods. Enhanced stability. Conjugation with vitamins may also protect nucleic acids from degradation within the biological environment, enhancing their stability and bioavailability. Challenges. Conjugation and synthesis. The chemical conjugation of vitamins to nucleic acids or vectors can be complex, potentially affecting the biological activity of both the vitamin and the nucleic acid. Limited tissue specificity. While vitamins can target specific receptors, the broad distribution of these receptors in the body might limit the tissue specificity of the delivery system. Endosomal escape. For receptor-mediated endocytosis pathways, ensuring the efficient escape of the genetic material from endosomes into the cytoplasm remains a critical challenge to achieving effective transfection. Lipids and fatty acids. Cholesterol enhances incorporation into lipoprotein particles for improved circulation and uptake by cells. Cholesterol, a key component of cellular membranes, has been widely utilized in the field of gene delivery and transfection due to its ability to enhance the cellular uptake and membrane integration of therapeutic nucleic acids. The inclusion of cholesterol in the formulation of transfection agents, such as liposomes and lipid nanoparticles, LNPs, significantly improves the delivery efficiency of genetic material into target cells. Here's a detailed overview of how cholesterol is used in transfection. Role of cholesterol in transfection. 1. Membrane fluidity and fusion. Cholesterol is critical for maintaining the structural integrity and fluidity of cell membranes. By incorporating cholesterol into transfection vectors, it's possible to facilitate membrane fusion between the delivery vehicle and cellular membranes, enhancing the internalization of the nucleic acid cargo. 2. Stabilization of delivery vehicles. Cholesterol helps stabilize lipid-based delivery vehicles, such as liposomes and LNPs, by preventing their aggregation and fusion in the bloodstream. This stability is crucial for maintaining the bioavailability of the genetic material until it reaches the target cells. 3. Enhancing endosomal escape. Once inside the cell, the successful release of nucleic acids from endosomes into the cytoplasm is a critical step for effective gene expression. Cholesterol can aid in destabilizing the endosomal membrane, promoting the escape of the nucleic acid cargo. Applications in transfection. Gene therapy. Cholesterol-modified delivery systems are employed in gene therapy to transport DNA plasmids, oligonucleotides like siRNA and antisense oligonucleotides, and mRNA to specific cells or tissues, aiming to correct genetic disorders, modulate gene expression, or induce protein production. CRISPR-Cas9 genome editing. The delivery of CRISPR-Cas9 components guide RNA and Cas9 nuclease to targeted cells for genome editing applications, benefits from the enhanced delivery efficiency provided by cholesterol incorporation. Vaccine development. Cholesterol is also utilized in the formulation of mRNA vaccines, where it plays a critical role in the stability and delivery efficiency of LNPs carrying the mRNA vaccine. Advantages. Improved transfection efficiency. Cholesterol enhances the interaction between the delivery vehicle and cell membranes, increasing the rate of cellular uptake and the overall efficiency of transfection. Reduced cytotoxicity. By stabilizing delivery vectors, cholesterol can help reduce the cytotoxic effects associated with some transfection agents, promoting safer gene delivery. Versatility. Cholesterol can be used in various transfection vectors and across different types of nucleic acids, making it a versatile component in gene delivery applications. Challenges. Optimization of cholesterol content. The proportion of cholesterol in delivery vectors must be carefully optimized to balance membrane fluidity and stability without compromising the release of the nucleic acid cargo. Cell type specificity. While cholesterol can enhance delivery efficiency, targeting specific cell types may require additional modifications or targeting ligands to ensure precise gene delivery. Regulatory considerations. For therapeutic applications, the safety, efficacy, and quality of cholesterol-containing delivery systems must meet stringent regulatory standards. DSP-PEG, 1,2, distearoyl sen-glycerol-3, phosphoethanolamine N, methoxypolyethylene glycol. Used in lipid nanoparticles, LNPs, for siRNA delivery, improving stability and circulation time. DSP-PEG, or 1,2-distearoyl N-glycerol-3 ion, phosphoethanolamine N-methoxypolyethylene glycol, is a lipid molecule that plays a crucial role in the formulation of lipid-based delivery systems for transfection. This compound combines a phospholipid, DSPE, known for its role in membrane structure, with polyethylene glycol, PAG, a polymer used to increase solubility and biocompatibility. The integration of DSPE-PEG into lipid nanoparticles, LNPs, and other lipid-based vectors has significantly advanced the field of gene delivery by enhancing the stability, biocompatibility, and targeting capabilities of these systems. Here's an overview of how DSPE-PEG is utilized in transfection. Role of DSPE-PEG in transfection. Stabilization of lipid nanoparticles. DSPE-PEG contributes to the structural integrity of LNPs and other lipid-based vectors, preventing aggregation in biological fluids. This stabilization is essential for the systemic delivery of genetic material. Stealth properties. The pegylation of lipid particles, that is, the addition of PEG, creates a hydrophilic barrier around the particle, which reduces opsonization, the process by which particles become coated with opsonins for phagocytosis, and delays clearance by the mononuclear phagocyte system, MPS. This stealth property extends the circulation time of LNPs, allowing them to reach their target cells more effectively. Targeted delivery. The distal end of the PEG chain can be conjugated with ligands or antibodies that target specific receptors on the surface of cells, enabling the targeted delivery of the LNP cargoes to specific tissues or cell types. Advantages: enhanced stability and circulation time. The inclusion of DSPE-PEG in delivery vectors improves their stability and circulation, enabling them to reach target tissues more effectively. Reduced immunogenicity. Pegylation with DSPE-PEG can mask the vector from the immune system, reducing immunogenicity and potential side effects. Versatility and modifiability. The pegylated end of DSPE-PEG can be easily modified with targeting ligands, allowing for the customization of LNPs for specific applications and targets. Challenges. Pegylation dilemma. While pegylation helps evade the immune system and extend circulation time, it can also reduce the cellular uptake and endosomal escape of the LNPs, a phenomenon known as the pegylation dilemma. Potential for anti-PEG antibodies. Repeated administration of pegylated formulations can lead to the generation of anti-PEG antibodies, potentially reducing efficacy and safety in some patients. Optimization. The optimal density and length of PEG chains require careful optimization to balance stability, stealth, and delivery efficiency. Spermine and other polyamines can condense DNA and improve packaging and delivery efficiency. Spermine and other polyamines, such as spermidine and putrescine, are organic cations that play crucial roles in cellular processes, including DNA stabilization, RNA transcription, and protein synthesis. In the context of transfection, polyamines are used to enhance the delivery of nucleic acids into cells. Their polycationic nature allows them to condense negatively charged DNA or RNA molecules, facilitating the formation of complexes that can more easily interact with cell membranes and be internalized by cells. Here's a comprehensive overview of how spermine, spermidine, and other polyamines are utilized in transfection. Mechanism of action: DNA and RNA condensation. Polyamines can neutralize the negative charge of nucleic acids, leading to the condensation of DNA or RNA. This condensation is critical for forming compact complexes that can be more efficiently taken up by cells. Enhancement of cellular uptake. The condensed nucleic acid polyamine complexes can interact more readily with cell membranes, partly due to the reduction in charge repulsion. This interaction facilitates the cellular uptake of the genetic material, either through endocytosis or other mechanisms. Protection against degradation. Polyamines can protect nucleic acids from degradation by nucleuses in the extracellular environment, thereby increasing the stability of the genetic material before it reaches the target cells. Advantages: non-viral. Polyamine-based transfection offers a safer alternative to viral transfection methods, reducing the risk of immunogenicity and insertional mutagenesis. Efficiency. The ability of polyamines to condense nucleic acids and facilitate their uptake by cells can lead to higher transfection efficiencies in some cell types, especially those that are difficult to transfect using other methods. Simplicity and versatility. Polyamine-based transfection protocols are generally simpler and can be applied to a wide range of nucleic acids and cell types. Challenges: cytotoxicity. High concentrations of polyamines can be cytotoxic to cells, limiting the maximum amount that can be used for transfection. Variable efficiency. The efficiency of polyamine-based transfection can vary significantly between different cell types and conditions, requiring optimization for each specific application. Endosomal escape. While polyamines can facilitate the uptake of nucleic acids into cells, ensuring that these molecules escape from endosomes into the cytoplasm to exert their biological effects remains a challenge. Polymers. Polyethylene glycol, PEG, used to increase solubility, reduce immunogenicity, and extend the half-life of oligonucleotides in the bloodstream. Polyethylene glycol, PEG, is a versatile biocompatible polymer widely used in pharmaceutical formulations, including transfection agents, to improve the delivery of nucleic acids into cells. The use of PEG in transfection is primarily focused on enhancing the efficiency and safety of non-viral gene delivery systems, such as lipid nanoparticles, LNPs, lipoplexes, and polyplexes. Here's a detailed overview of how PEG is utilized in transfection processes. Mechanism of PEG action in transfection. Stabilization of delivery vectors. Pegylation. The process of attaching PEG polymers to another molecule helps stabilize lipid and polymer-based vectors by increasing their solubility and preventing aggregation in physiological conditions. This stability is crucial for maintaining the integrity of the delivery vectors as they navigate through the body to reach target cells. Reduced opsonization and prolonged circulation. Pegylation provides a stealth characteristic to delivery vectors, shielding them from the immune system. By reducing protein absorption, opsonization, and recognition by the mononuclear phagocyte system, PEG extends the circulation time of these vectors, allowing for improved accumulation in target tissues. Tunable release and targeting. The attachment of PEG to delivery vectors can be customized to modulate the release rate of the nucleic acids and to include targeting ligands. By adjusting the molecular weight and density of PEG, as well as incorporating ligands at the distal end of PEG chains, it's possible to achieve targeted delivery to specific cell types or tissues. Advantages: biocompatibility and low toxicity. PEG is non-toxic and biocompatible, making it an ideal component for use in medical applications, including transfection. Increased efficacy. By enhancing the stability and circulation time of delivery vectors, PEG can increase the overall efficacy of the transfection process. Versatility. Pegylation can be applied to various delivery vectors and is suitable for delivering different types of nucleic acids to a wide range of cell types. Challenges. Potential for reduced cellular uptake. The presence of PEG can sometimes hinder the interaction between delivery vectors and cell membranes, potentially reducing the efficiency of cellular uptake. PEG dilemma. While pegylation helps evade immune detection, it can also limit the endosomal escape and release of nucleic acids within target cells, which is essential for their therapeutic effect. Immunogenicity of PEG. Repeated administration of pegylated formulations can lead to the production of anti-PEG antibodies in some individuals, reducing the efficacy of treatment and potentially leading to adverse reactions. Polylactic co-glycolic acid, PLGA, biodegradable polymer used in nanoparticle formulations for controlled release. Polylactic co-glycolic acid, PLGA, is a biodegradable and biocompatible copolymer widely used in the field of drug delivery, including the transfection of nucleic acids into cells. Due to its favorable properties such as biodegradability, tunable degradation rate, and safety profile, PLGA has garnered significant interest for its application in non-viral gene delivery systems. Here's a comprehensive overview of how PLGA is utilized in transfection. Basic properties of PLGA. Biodegradability. PLGA degrades into lactic acid and glycolic acid, which are naturally metabolized by the body, reducing the risk of long-term toxicity. Biocompatibility. PLGA is well tolerated by the human body, making it suitable for medical applications, including implantable devices and drug delivery systems. Versatility. The degradation rate of PLGA can be tailored by adjusting the ratio of lactic acid to glycolic acid, allowing for controlled release of encapsulated agents. Mechanism of PLGA in transfection. Encapsulation of nucleic acids. PLGA nanoparticles can encapsulate nucleic acids, DNA, RNA, siRNA, etc., protecting them from enzymatic degradation and facilitating their delivery into cells. Cellular uptake. The nanoparticles are taken up by cells through endocytosis, a process that can be enhanced by modifying the surface of PLGA nanoparticles with targeting ligands or cell-penetrating peptides. Controlled release. Once inside the cell, the PLGA nanoparticles degrade, releasing the encapsulated nucleic acids in a controlled manner, which can then exert their therapeutic effect by modulating gene expression or correcting genetic defects. Advantages: enhanced stability and protection. PLGA provides physical protection to nucleic acids against enzymatic degradation, enhancing their stability in the biological environment. Controlled release. The degradation rate of PLGA can be tuned to achieve a controlled release of the encapsulated nucleic acids, allowing for sustained gene expression or silencing over time. Safety and biocompatibility. The biodegradable and biocompatible nature of PLGA minimizes toxicity and adverse immune responses, making it suitable for in vivo applications. Challenges: efficiency of transfection. The efficiency of PLGA-based transfection systems can be lower than viral vectors, necessitating further optimization to improve delivery and gene expression levels. Endosomal escape. Ensuring that the released nucleic acids escape from endosomes into the cytoplasm is crucial for effective transfection, but can be challenging with PLGA nanoparticles. Scale-up and reproducibility. Producing PLGA nanoparticles with consistent quality and in large quantities can be challenging, affecting the scalability of this approach. Carbohydrates. Mannose. Targets mannose receptors on macrophages and dendritic cells, useful for immunotherapy applications. Mannose, a simple sugar, plays a unique role in transfection and gene delivery strategies, particularly due to its ability to target and enhance delivery to specific types of cells that express mannose receptors. These receptors are primarily found on the surface of macrophages, dendritic cells, and some types of cancer cells, making mannose an attractive ligand for targeting and delivering therapeutic agents to these cells. The use of mannose in transfection leverages its natural biological interactions to improve the efficiency and specificity of gene delivery systems. Here's a comprehensive overview of how mannose is utilized in transfection. Mechanism of mannose in transfection. Targeted delivery. Mannose-modified vectors exploit the mannose receptor-mediated endocytosis pathway. These receptors have a high affinity for mannose and related sugars, enabling targeted delivery of the vector to cells that overexpress mannose receptors. Enhanced cellular uptake. Upon binding to mannose receptors, the mannose-modified transfection vectors are internalized into the cells via receptor-mediated endocytosis. This targeted approach helps to increase the efficiency of cellular uptake of the delivered genes or nucleic acids. Immune system activation. In addition to direct targeting of specific cell types, mannose can also play a role in activating the immune system. Mannose receptors are part of the pattern recognition receptors, PRRs, on immune cells, and their engagement can stimulate immune responses, which can be beneficial in vaccine delivery and cancer immunotherapy. Advantages: targeted delivery. The primary advantage of using mannose in transfection is its ability to target specific cell types, increasing the specificity and efficacy of the gene delivery while minimizing off-target effects. Reduced toxicity. Targeted delivery to specific cells reduces the amount of vector required for effective transfection, potentially decreasing the toxicity associated with higher doses of transfection agents. Enhanced immune response. In applications where an immune response is desirable, such as in cancer therapy or vaccination, mannose's ability to stimulate immune cells can be beneficial. Challenges: limited target scope. The effectiveness of mannose targeting is limited to cells that express mannose receptors, which may restrict its applicability to certain therapeutic areas. Complex synthesis. The chemical synthesis and conjugation of mannose to vectors can be complex, requiring careful optimization to preserve the targeting functionality of mannose while maintaining the biological activity of the nucleic acids. Endosomal escape. Once internalized, ensuring that the genetic material escapes from the endosomes into the cytoplasm is crucial for successful transfection and remains a challenge for mannose-targeted delivery systems. Hydrophilic sugars can improve solubility and biocompatibility of oligonucleotide conjugates. The use of hydrophilic sugars in transfection leverages their natural biocompatibility and targeting capabilities to enhance the delivery of nucleic acids into specific cell types. These sugars, including mannose, galactose, and fucose, can be conjugated to vectors or delivery systems to improve transfection efficiency, specificity, and safety. The strategy primarily revolves around the interaction between these sugars and specific sugar receptors, such as lectins, on the surface of target cells. Here's a complete overview of how hydrophilic sugars are utilized in transfection. Mechanism of action. Targeted delivery. Hydrophilic sugars can target specific receptors on cell surfaces. For instance, mannose targets mannose receptors on macrophages and dendritic cells. Galactose targets Asia low-glycoprotein receptors on hepatocytes, and fucose may target selectins. This receptor-mediated targeting facilitates the uptake of the sugar-modified vectors into specific cell types. Enhanced cellular uptake. The binding of sugar-conjugated vectors to their corresponding cell surface receptors triggers receptor-mediated endocytosis, thereby enhancing the cellular uptake of the encapsulated or conjugated nucleic acids. Reduced immunogenicity and toxicity. The biocompatibility of sugars can minimize the immunogenic response and cytotoxicity often associated with synthetic vectors, making transfection safer for therapeutic applications. Advantages: specificity. The ability to target specific cell types through their sugar receptors increases the specificity of transfection, reducing off-target effects and enhancing therapeutic efficacy. Biocompatibility. Hydrophilic sugars are generally biocompatible and can reduce the immunogenicity and toxicity of delivery vectors. Versatility. Different sugars can be used to target different cell types, making this approach versatile across a range of therapeutic areas and applications. Challenges. Complexity of vector modification. The chemical conjugation of sugars to vectors or nucleic acids requires careful optimization to maintain the targeting ability of the sugars while preserving the biological activity of the nucleic acids. Endosomal escape. After receptor-mediated endocytosis, ensuring that the delivered nucleic acids escape from the endosomes into the cytoplasm to exert their therapeutic effect is crucial and remains a challenge. Limited target range. The effectiveness of this strategy is limited to cells expressing the specific sugar receptors, potentially narrowing its applicability to certain cell types or conditions. Metallic and inorganic molecules. Gold nanoparticles can be conjugated with oligonucleotides for improved delivery and as part of therapeutic or diagnostic systems. Gold nanoparticles, AONPs, have emerged as a powerful tool in the field of transfection, offering a novel approach to deliver genetic material into cells with high efficiency and specificity. Their unique physical and chemical properties, including size, surface area, and the ability to easily modify their surface with various ligands, make them an attractive vector for gene delivery. Here's a comprehensive overview of how gold nanoparticles are utilized in transfection. Basic properties of gold nanoparticles: size and shape. The size and shape of AONPs can be precisely controlled during synthesis, affecting their cellular uptake and biodistribution. Sizes typically range from 1 to 100 nanometers in diameter. Surface modification. AONPs can be functionalized with a variety of molecules, including DNA, RNA, siRNA, peptides, and polymers, enabling targeted delivery and improved compatibility with biological systems. Optical properties. AONPs have unique optical properties, such as strong surface plasmon resonance, which can be utilized for imaging and tracking the transfection process. Mechanism of AONP in transfection. Nucleic acid conjugation. Nucleic acids can be directly attached to the surface of AONPs through covalent bonds or electrostatic interactions. This conjugation protects the genetic material from enzymatic degradation and facilitates its delivery into cells. Cellular uptake. The modified AONPs are internalized by cells predominantly through endocytosis. The efficiency of uptake can be enhanced by optimizing the size, surface charge, and surface coating of the nanoparticles. Endosomal escape. Following internalization, the AONPs can facilitate the escape of the nucleic acids from endosomes, a crucial step for successful transfection, allowing the genetic material to reach the cytoplasm or nucleus where it can exert its effect. Advantages. High stability and protection. AONPs protect nucleic acids from nuclease degradation, enhancing the stability of the genetic material in biological environments. Targeted delivery. Surface modifications allow for the targeting of specific cell types or tissues, increasing the specificity and efficacy of transfection while minimizing off-target effects. Non-viral. As a non-viral vector, AONPs avoid many of the safety concerns associated with viral transfection methods, such as immunogenicity and insertional mutagenesis. Challenges. Cytotoxicity. While generally considered biocompatible, the potential cytotoxicity of AONPs depending on their size, dose, and surface chemistry remains a concern and requires careful evaluation. Endosomal escape. Efficient endosomal escape is crucial for successful transfection, but can be challenging to achieve with AONPs, necessitating further optimization. Regulatory and scale-up. The production, characterization, and regulatory approval of AONP-based transfection systems can be complex, impacting their clinical translation and commercialization. Quantum dots, used in research settings for tracking and imaging purposes. Quantum dots, QDs, are nanometer-scale semiconductor particles that exhibit unique optical and electrical properties, making them highly valuable in various scientific and medical applications, including transfection. Their distinct size-dependent fluorescence emission, high photostability, and the ability to conjugate with biological molecules have paved the way for their use in cellular imaging, tracking, and the delivery of genetic material into cells. Here's a comprehensive overview of how quantum dots are utilized in transfection. Basic properties of quantum dots. Size and composition. Quantum dots typically range from 2 to 10 nanometers in diameter. They can be made from various semiconductor materials such as cadmium selenide, CSE, or indium phosphide, INP, each offering different optical properties. Fluorescence QDs can absorb and emit light of specific wavelengths, with the color of the light dependent on the size of the quantum dot. This property is used for labeling and tracking in biological applications. Surface modification. The surface of QDs can be chemically modified to attach biological molecules, including nucleic acids, DNA, RNA, proteins, and targeting ligands, enabling their use in targeted delivery and transfection. Mechanism of QD and transfection. Conjugation with nucleic acids. Quantum dots can be conjugated with nucleic acids either directly through covalent bonding or indirectly using linkers. This conjugation allows QDs to carry genetic material for delivery into cells. Targeted delivery and cellular uptake. By modifying the surface of QDs with specific ligands or antibodies, they can be directed to target specific cell types or tissues, enhancing the specificity of the transfection. Cells internalize these conjugated QDs through mechanisms such as receptor-mediated endocytosis, endosomal escape and release of genetic material. Once inside the cell, the QDs must escape from endosomal compartments to release their nucleic acid cargo into the cytoplasm or nucleus, where it can exert its intended effect. Advantages. High photostability. The photostability of QDs allows for long-term imaging and tracking of transfection processes and gene expression in living cells. Multiplexing capability. Different sizes of QDs emit light at different wavelengths, allowing multiple genetic constructs to be tracked simultaneously within the same cell or tissue. Targeted delivery. Surface modification of QDs enables the targeting of specific cell types, improving the efficiency and specificity of transfection. Challenges. Cytotoxicity. The potential cytotoxicity of quantum dots, especially those containing heavy metals like cadmium, is a significant concern that limits their use, necessitating the development of safer, less toxic QDs. Endosomal escape. Efficient release of the nucleic acid cargo from endosomes into the cytoplasm or nucleus remains challenging, affecting the efficiency of gene delivery. Regulatory and safety concerns. The long-term stability and potential bioaccumulation of QDs pose safety and environmental concerns, impacting their clinical and commercial use. Advantages and challenges. The conjugation of oligonucleotides with these ligands can significantly enhance their therapeutic potential by improving delivery to the target site, increasing cellular uptake, and minimizing off-target effects. However, challenges such as the complexity of conjugate synthesis, potential toxicity, immunogenicity, and ensuring the stability of the conjugate in biological systems must be carefully managed. Future directions. Continued innovation in ligand discovery and conjugation chemistry is expanding the possibilities for oligonucleotide therapeutics. Advances in targeted delivery, coupled with a deeper understanding of disease mechanisms at the molecular level, promise to yield more effective and safer treatments for a wide range of conditions. Stay curious, stay innovative. I'm Luke McLaughlin. If you would like to learn more about this topic, consider reading our full-length articles on biotechnologyreviews.com, or consider our podcast series on YouTube or Spotify at our Biotechnology Reviews channel. Our articles are also available on our LinkedIn newsletter, Biotechnology Reviews.